All right, you guys, here we go. So different chapters are talking about different parts of the reflex arc. And I told you when we first learned reflex arc that we're going to refer to them in every system or chapter that we're studying. So this side of the sensory reflex is your chapter 10. So what we're talking about in this chapter is all about receptors. Where do we have them? What kind of receptors we have, either general or special. And obviously the special are all in our head, right? So your eyes are receptors, your tongue is a receptor because it has the gustatory cells. Your nose has receptors, which are the olfactory receptors, and your ears have receptors for hearing and for equilibrium. So that's what we're doing. Now, when we studied chapter 9, it was pretty much this one. Is that side, because we were learning how the effectors in the autonomic nervous system were responding, okay? So if we have time at the end of our pre-lab, I'm gonna go over that a little bit more so that you have it clear. When we study, when we're gonna study chapter 12, we're also gonna be talking about that motor effector type of reflex on that side. So, but for now, chapter 10, it has all to do with sensory, the sensory side. How do we detect it? How do we send it to the interpretation center? And we're not talking about how we are resolving that, but it's mostly that reception and that sensory information going up, okay? All right, so I'll go back to the PowerPoint with our vision. And we have several little virtual simulations. So the first one that you have is called the accommodation of the lens. So in the anatomy of the eye, we have the lens, which are structures made out of protein layers. In fact, if you've done 201, well, if you did it virtually, no, but if you did 201 on campus, we dissect a cow eye and you can literally peel the lens like an onion. So that's how these proteins are arranged in many, many different layers. So the lens, it's that refracts the light. And in order for us to really form the image on the retina, which is the normal type of vision, the lens is attached to ciliary muscles on top and at the bottom, and they have to either get rounder or they get flatter. And that's what accommodation is all about. Testing accommodation also gives the eye doctor an idea of how good your oculomotor nerve is the cranial number 3. Because accommodation is almost like when you use a microscope and you're trying to focus with the fine adjustment knob so that you can really see a sharp image. So that's what really accommodation is all about. And when you find the object and the object is clear, that's what is called near point of focus. So the distance when you can see something very clear or sharp is called near point of focus. So the typical eye exam that some of you maybe have done is when looking at a chart, and we're going to have that too in one of the virtual simulations. And that chart is called Snellen chart. And this is where you can see sharp objects at about 20 feet away. So it requires your lens to do this kind of adjustment and accommodation so that you can focus not only on the retina, but on the fovea centralis, which is the most accurate vision. This is where we have a lot more photoreceptors, and that's when you have the most sensitivity to focusing on that point of the retina. So these lenses, like I mentioned, they are attached in with ciliary muscles, and these muscles are made out of protein as well, and they get a little lax, or they don't work that well as people age. So most of the times, somebody that is over 40 will need reading glasses, okay? So here is a little bit of the anatomy of the eye. So what you have here is the eyeball. The eyeball, if you notice, the outermost region of the eyeball is called the sclera, is the outermost layer, we'll say. Then the next one over that you see here with little dots, that's called the choroid layer or choroid coat. And the little nuclei that you see there is because this is highly, highly vascularized, meaning that it has a lot of blood vessels, so that the photoreceptors, which are on the innermost layer, which is the retina, so this one right here, so the photoreceptors get the oxygen and the nutrients that they need. Now, the, let me see, question? Okay, no problem, Danny. All right, so those are the three layers of the eye: the sclera, the choroid coat, and the retina. The retina is the thinner innermost layer, and it is in the retina that we have the photoreceptors, right? So those are the ones detecting the information, and then that information is sent to other cells and ultimately to what they're called ganglion cells. So ganglion cells are, let's see if I write it here, ganglion cells. So ganglion cells are the ones which axons are going to form the optic nerve. Okay, so the optic nerve is right here. Okay, and you can see too that there's a lot of blood vessels right there. You have a artery, you have veins, so red arteries, blue veins. So you can tell that these arteries and veins are innervating the whole retina. All these little blood vessels that you see here are extremely important because that's how our retina cells get the nutrients and the oxygen. Now, if you have a big blow to your head, then the retina can get detached from that middle layer. And that's a problem because then you cut the circulation to the retina, and then the photoreceptors can start to die without oxygen, without blood supply, and that can lead to blindness, right? So there's times that you have partial detachment of the retina, and that it's not as serious if it's taken care of very fast. They can do laser surgery and they reattach it back to the choroid coat. Or it could be a total detachment of the retina, and that's more serious. You can attach it with laser, but sometimes not the whole retina is attached again, and some parts of the retina may be altered. So boxers, for example, are at a higher risk of retina detachment. And in fact, it has happened in some fights where they had to stop the fight and they have to rush the athlete into surgery right away, so to preserve their eyesight. So that's a little bit of the anatomy of these three layers. Now, if you notice, the sclera, which is the white part of the eye, goes all the way to the front, and within this region in the front, the sclera becomes a little skinnier or thinner. In fact, it changes epithelium, and that's what is called the cornea. So the cornea right here is the clear window of the eye. That's what it's called, right? The sclera is made out of dense white fibrous connective tissue. So if you took 201, you know what I'm talking about. Is a lot of collagen fiber. So it's a very strong type of tissue and very dense. But when we get to the front of the eye, to the cornea is made out of simple stratified squamous epithelium. So a very different kind of epithelium tissue. And that's the reason why if anybody has a scratched cornea, that heals and very fast because it's the same epithelium that we have on our skin. stratified squamous epithelium, right? The difference between this epithelium and the skin is that on our skin, we also have keratin. So it's keratinized stratified squamous epithelium. But in the cornea, we don't have keratin. But if you have a scratch cornea, like my daughter is a 4th grade teacher, and one day, one of the students, when we were face-to-face, came to give her a paper and stuck the paper in her eye. It was such a coincidence, right? So she started, like, it was blurry, she started crying, and the next day she had to go to the doctor because it was really inflamed and swollen, but it was a scratch cornea, right? It was a paper cut, same as you will get it in your skin. So it is very uncomfortable, but it heals very fast because it's the same as having a scratch on your skin. It heals. Remember that stratified squamous epithelium, we're constantly shedding off the first layer, the outer layer. So the cornea heals very fast. You need some drops, couple of days, and you start healing. So that's what the cornea, and it's clear because we need to allow for the light to go through. So you can see that the light goes through the cornea, and then we have here the iris. The iris is the color portion of the eye, and in the center we have the pupil. Right? Now, right behind it, we have the lens. And so you can see here the lens, and it's held by these little suspensory ligaments that are attached to these ciliary muscles. So this whole structure here, the suspensory ligaments from the ciliary muscles and the lens are the ones that are doing the movement on making the lens flatter or making it rounder. So going back to accommodation, if we need to look at something that is distant, the lens is going to be flatter. So the suspensory ligaments are going to pull a little more and the lens become flatter than before. And on the other side, on the right, if you need to kind of like look at your watch, which is near vision, the lens becomes a little rounder. So you can see that the ciliary muscles are going to contract, moving everything closer, and that makes the lens rounder. So that's how we accommodate. Now, all the time, what we want to do is to form the image on the retina, right? Because that's where we have the photoreceptors. Questions so far? We're good? I gave you a little bit of the anatomy. So this is what I was talking about. I don't know if you noticed, your parents or grandparents, at one point in our lives, and that's why I have glasses because I'm looking near my computer, but at one point we start putting our reading material farther away from our eyes, right? So that happens because you are not able to do the accommodation that good. And instead of being able to read at a normal distance, the arms become really long and you put things away trying to focus on the retina. Because if you don't do that, you couldn't focus on the retina and you couldn't see sharp. So it's very important for reading, right? That we can see very sharp. So that's about 40 to 45. You can see how the near point of focus starts becoming increased, right? And then it gets to a point that your arms are not that long anymore. And that's when you need to buy reading glasses. And those are the ones that we buy in CVS, anywhere, you know, they're not prescription, they're just readers, they call them. So you can see here that when you are in your 20s or your 30s, you're pretty good. This is in centimeters, but by the time you reach 40, 50, or even 60, you know that your arms are not this long anymore, so you can't push things away. So that's when you need your readers. So that's the first virtual simulation that we're going to do, which is the accommodation, finding that near point of vision. The next one you're going to do is called the blind spot demonstration. So the blind spot is a little bit away from the optic nerve. Not away, sorry. The fovea centralis is a little bit away of the optic nerve. Now, where the optic nerve meets the retina, that's what we call the optic disc. So optic disc is called a blind spot because it doesn't have photoreceptors. Now, fovea centralis, which is around this region here, that's when you have the most accurate vision because that's when the photoreceptors are the most sensitive, okay? So this one, this simulation will try to find the blind spot. And of course, we have one on each eye, right? Because we have two eyes, two optic nerves, and two blind spots. So again, this is your oculomotor, cranial nerve number 2, optic nerve, cranial nerve number 2. Oculomotor is number 3, is the one that helps us move our eye. So they're both involved. So in this simulation, you're going to see that the patient is going to cover one eye, and we're going to try to find or visualize the blind spot with a chart that is going to be at a normal distance. And then that chart is going to start getting closer to the patient. And at one point, the dot on that chart is going to disappear. So when it disappears and we measure the distance, that's what it means that you found the blind spot. Now, because we have two blind spots, when we don't have our eyes covered, one eye makes up for the other eye's blind spot and vice versa, okay? So it is important too, because they do it in a routine examination, or if they know that somebody has suffered some injury, they want to make sure that the blind spot is there, and they want to find it doing that. So first they cover one eye and they find the blind spot on the other eye, and then you cover the next eye and you find the blind spot on the second one. So a blind spot is located on the lateral of each eye, okay? Lateral side. Now, this one is one of my favorites when we used to do this one in face-to-face labs, because not that I wanted to diagnose anybody being colorblind, but a lot of students would realize, oh my God, I'm colorblind. So colorblind has to do with cones and rods, right? Those are the two photoreceptors that we have. So the rods are the ones that are determining the whites and the blacks and any gray in between. And of course, they're determined to be more active during dim light. And cones are the ones that detect color. So we have three parts or three cones, and the three are red, blue, and green, so they can detect that colored light wavelength. And then combined with the black and white of the rods, we can make up all the colors in the spectrum, right? So we have a test that is done. It's called the Ishihara plates, and it's Mr. Ishihara designed this. Uh-oh, my pool guy is here and my dog is gonna start going nuts, you guys. Let me close the door. You still hear her, but not a little muffled. All right. So Mr. Ishihara designed these plates, they're called, where you have to identify a number or a shape. And for some people that are colorblind are not able to do this. So there are different kinds of color blindness. It could be the red-blue or the green-red color blindness. Green-red is more common. And of course, it's more common in males because color blindness is a genetic trait that is carried in the X chromosome. So females can be carriers because we have two X's, so it's not as common. But remember that one of our X's in the female is inactivated during development. So anything on that X is not going to be showing in the organism. But in males, whatever they have in that X is going to be expressed. So if they have that mutation for color blindness, they're going to be colorblind. So the results of the little Ishihara plates, these are the plates, and you can see, I'm sure that you can tell that this is a 12 in this one, right? Salita is asking, is this why that can make out different shades of colors? Yeah, it could be. So how about on this one, on the second one, right? I mean, I had a hard time, but I can tell that it's an 8. I hope you all see an 8. And then the third one here is a 16. And then the last one here is a 96, right? So obviously you see here, this is the results of the little virtual simulation. When you don't have any color deficiency, you can identify a single number. And when you're green colorblind and you don't know if it's a 3 or whatever it is, you don't really see this. And in here, that person will only see a 9. And if you have red color blindness, they can do that one, but then the middle one, 3 or no, no, and this person can see a 6. So this is a way of obviously determining if you're colorblind. And think about some of the professions that it will be a problem to be colorblind, right? So an ambulance driver, right? So problem, if you can't tell if is the green light, the red light, or whatever light you have to go through or not, right? Pilot, yes, Salita, a pilot, yeah, you have to see the lights, right? When you're landing or leaving. What else? What else could it be? How about a painter? You hire a painter for your house and you say, I want green color, and they paint the house red. That would not be good, right? So yeah, there are certain professions. Oh, electrician is another one. Electrician that doesn't know the red color cable, right? And that one is the hot wire. So that's a problem, right? So they're connecting things the wrong way. So I hope that somebody that is colorblind doesn't pick one of those professions. But I have a funny story. My daughter, when they were still, I think they were still living at home. Yeah. So my kids just turned 38 yesterday. So they're old people now. But when they were still at home, I remember in this room, when I have my office now, we used to have it as a playroom for them. They had video games, and we had a couch. And my daughter one day, we're downstairs, and she says, oh, yeah, I think I left it on the green couch upstairs. And I look at her and I go, we don't have a green couch. What are you talking about? She goes, yeah, the one upstairs in the playroom. I said, that couch is blue. So then we realized that she has some kind of color blindness, because for the longest time, and I think we had that couch for like 10 years, she thought it was green and really blue, but it was that blue, you know, like kind of like in between. So she's not totally, but I think some of her photoreceptors are not that great, I would say. All right, so the next little simulation that you're going to do is the pupillary reflex test. So this one has to do with your pupil, right? And the pupil is the opening that we have in the iris, and the iris has these sphincter little muscles, and they have radial going out like this, little fibers going out on every direction. And then we have circular little fiber muscles there. So that's what allows for pupil constriction or pupil dilation, right? So why do we have that? We have it is to protect our brain really from getting a lot of light in, and protect our photoreceptors. When you shine a really bright light on your eyes, that's a lot of light. So you want to protect your retina, your photoreceptors, so the pupil constricts. And when there's not a lot of light, imagine you walking in a dim light, or even a dark room. it takes a while, right, for us to get used to, and sometimes you start seeing things. And this is because your pupil will dilate and it will open up as much as possible to get as much light there is in that room as possible so you can see. And I'm sure you've seen some of the medical shows out there that if somebody walks into the ER, immediately they do that little pupillary reflex so that they want to see if they are constricting the pupil, because that will give you also an indication of concussion. So any hard hit to the head, any blow to the head could damage that reflex. So, and this is what I just said, when you walk into a dim room, you're going to dilate. So this is the sympathetic reflex. And when you go into a bright light, you're going to constrict, which is a parasympathetic reflex. And in this case, if you remember anything about parasympathetic, is the opposite, right? You would think that constriction will be sympathetic, but it's not. And how do you determine that? How do you remember which one is a sympathetic or which one is a parasympathetic event, right? So I'm going to tell you for this one, just for this one. Sympathetic, you dilate your pupil because you want to see everything that is happening when you are in the flight or fight response, right? So think about running in a dark alley from a guy with a knife. You want to have very dilated pupils so you can see where you're going and you can get away from danger right away. So that's how we can determine, is it sympathetic or is not? Yeah, it's sympathetic, right? Because you want to see everything that is coming at you when you are in that adrenaline rush type of flight or fight. So you have that these little fibers around the pupil are innervated, and that means the nerves that are controlling these little fibers is again the oculomotor nerve. So anything that has to do with moving or doing constriction dilation, it has to do in the eyes, it has to do with the oculomotor. Motor means moving, right? Optic nerve is cranial nerve number 2, oculomotor is cranial nerve number 3. So again, when you are looking at things 20 feet or closer, your accommodation works also with the pupillary reflex because you want to make sure that you have the right amount of light entering the eye and that you're focusing on that part of the retina, which is the fovea centralis, right? Fovea centralis is a little bit away from the optic nerve area, and that's when your photoreceptors are the most sensitive. So in order to really see well and sharp, we have to do both. We have to do accommodation, which was our lens, and also we have to do the pupillary constriction or dilation, allowing for more or less light to come in. So this is a picture of your answers in that simulation. So you can see here how when this eye is being shown with a really bright light, how these little fibers, which we have circular and radial, so the radial are the ones going like that out and circular are around, you can see that the pupil is constricting and is becoming smaller. So that's parasympathetic, right? And then the opposite, when you don't have a lot of light, your pupil wants to dilate because you want to make sure that you get as much light as possible in order to form an image, trying to see something, and that's your sympathetic innervation. And remember, how do we remember which one is sympathetic or parasympathetic? Always know that in the sympathetic, the flight or fight makes you very apt to seeing everything you can. So you need more light or the most light that you can get inside to form the image on the retina. Questions so far on this? No, we're good. All right. So the next one that we're doing is visual acuity. And this is how accurate you are with your vision. So this is the typical test that we've all done when they show you the what is called the Snellen chart. And again, this is Mr. Snellen. He made up this. And this is when you are when you have good vision or visual acuity, you can see line 8 of the Snellen chart when you are 20 feet apart from the chart. So that's important for driving. I'm sure that you all done that when you have to look and you see, and they tell you read line 4, 5, 6, and they get to 8. And if you can't read it, it means you need glasses, right? So the whole point of this is that you should be able to form the image on the retina. You can see things very accurately if the image is formed in front of the retina, or if the image is formed back of the retina. And that has to do with your vision, right? So somebody that has perfect vision, you say, oh, that person has 20/20, okay? So 20/20 vision is when you can read that line number 8 from 20 feet of distance. Somebody that is 20/40, then that means that a person with normal vision can visualize that line from 40 feet away. But a person that cannot then is considered myopic or nearsighted. I'm nearsighted. All right, so normal vision, you are forming the image on the retina. So you can see here that all the rays of light are converging on the retina. Really? Right there. So you see how all these rays of light are coming through. They're going first through the pupil, right? Then the lens, and ultimately they're all converging on the retina. So that's what is considered normal vision. Normal vision is called emetropia. Emetropia. So here's your Snellen chart, and this is your line number 8, right? So if you're 20 feet away from this chart and you can read these letters, then you're considered to have 20/20 vision. This is normal. Now you could be really, really good, and from 20 feet, you can even read line 9. That would be that you are 20/15 vision. Better, right? Oops. And I haven't known that many people, but 2013, you can even read line 10, right? So that's when your image is not being formed on the retina. So we're gonna see what happens when your image is not formed on the retina. So here is myopia or nearsightedness. So now you can tell that the image is formed, let me see, in front of the retina, right? So you see that line that goes through, and all the light rays are converging, but instead of doing it on the retina, you're forming the image in front of the retina. And this is what happens. You can see that the Snellen chart is all blurry, right? You don't have that sharpness of focus. And imagine adding that to having presbyopia, which is after 40, it is a mess, right? So you need two different type of glasses. So I have my readers because I'm obviously over 40, and I also have my glasses for my myopia when I need to see from far away. So the next one is hyperopia. So now this is called farsightedness, and you can see where the image is formed now, right? It is behind the retina. So that's why it's called farsightedness. You're forming the image behind, all the light rays are converging, but they're not converging on the retina, they are converging behind it. So for both conditions, farsightedness or nearsightedness, you need corrective glasses, right? There's one more condition that you're not, I think, doing in the simulations, that is astigmatism. And astigmatism has to do with the shape of the lens. So that's pretty much genetic. This is how you come. And astigmatism can be the only thing that is wrong with some patients, or it could be in addition to, and I have it as well. I have everything, right? I need my readers, I have astigmatism, and I'm nearsighted. So the thing with the lens in an astigmatism is the shape of the lens. The shape of the lens is different or is not normal. So I'll show you how it is. I'm just gonna erase this. So it's the lens should be kind of like the same shape, right? On both sides like that. Astigmatism will be fatter on one side and flatter on the other. Or it could be fatter on the top and then skinny at the bottom. So instead of being two concave halves, you can have one side that is flattened or the bottom side is more flattened. So that is astigmatism. And there's also corrective lenses for that. All right, let's see what time is. Any questions on these little simulations that you're going to be doing or what you're going to be testing for? These are very common type of exams. If you go to the eye doctor, the Snellen chart, you do it every time you do driver's license test or things like that. So any questions on that? No? Oh, you're an optician, Salita. So you see that a lot. You do all these kind of tests?
I'm the one that orders the glasses and does all the measurements and stuff. So if you need glasses, I can usually tell you what you need off of the prescription and suggest what you should be getting.
Yeah, gosh, you remind me, I haven't been to my eye doctor in a while, so I probably need some updates.
I work for Costco, so it's cheap.
Oh yeah, I've gotten some lenses from Costco. That's good. Yeah. Anybody else that has any? I told you all my vision issues. Well, Karen, you have glasses. What do you have?
Oh, I'm very, very nearsighted.
See?
You can see about
Yeah.
5 inches in front of my face, and I have the astigmatism too.
Oh, great. Double whammy. All right. Okay, so I wanted to go over a little bit of chapter 9, but I'm gonna take a 10-minute break and we'll come back at 2. Okay, it's 1:49, so we'll come back at 2. Deal? All right. Okay, I'm back. So, Julie, you had questions on chapter 9.
Not on chapter 9, but on the test. I'm sorry if you covered it at the beginning of today's class. I was trying to figure out my headphones, but I know you mentioned there were a few incorrect answers. Well, let me back up. Will you be sharing the correct answers with us?
Sure, if you want.
I don't know if anybody else wants that, but I was curious about just the ones that I got wrong.
If you wanna go over those right now, I can do it. I'm sure that some people would like that too. If you wanna go over the ones you wanna know, that's
Sure.
fine.
The reason I'm asking is for question 16.
Yes, which was that question? Let me see if I can.
So solids that cannot freely pass through a membrane are said to be, and I thought the answer was automatically active, but I got it wrong.
Let me see. Let me just go to the exam so that I can look at it. Edit. Okay. And it says 16.
Yes.
Solute that cannot freely pass through a membrane are said to be osmotically active. What did you say?
Osmotically active.
I think
Miss
that's
Wanya,
the
hear
correct
me?
answer. Let me
Oh, it
check.
is. Okay.
Let me double check. All right. Huh. I can look at this. Questions. Okay. I'm just checking. 16. Yeah, they're osmotically active. And you know what? I have it wrong here. So that's one more that I need to check. Because I have the correct answer are selectively permeable, but that's the membrane.
Oh yeah, I got that one automatically wrong too.
Yeah. So I'll check those. Whoever got it right, I'll change it. You'll get a new little point there. Okay.
Thank you so much.
What else, Julie?
That was the only one that I was really perplexed by, and now I'm.
All right. Yeah, because selectively permeable refers to the membrane, not to the solutes.
Right.
Yeah. Exactly.
Yeah, and then I won't bore us with the other ones, but if I wasn't sure if you mentioned you were gonna put the correct answers up, or if they were gonna be visible at one point.
No,
eventually.
I didn't mention that. No,
Okay.
but I can do it for you. I'm just going to chapter 9. And did you have another question for chapter 9?
No, I did not.
Okay.
I
All right.
actually had one.
Okay.
Question 29, the most common neurotransmitter in the brain is, so I had a toss up between acetylcholine and GABA, so I wasn't unsure of that.
It's acetylcholine.
Okay, I thought so. I just second guessed myself.
Okay. Oh yeah, we do that all the time, right? I did a long time ago, I used to do a test taking strategies. And it was something that ASG was doing, and sometimes we would do it. And one of the things in multiple choice is always go with your first instinct. Never go back and change. That's one of the strategies. Your first instinct is most of the times right, correct. Now, true or false, it's 50-50. So I couldn't give you any strategy on that one. But multiple choice, yes, go with the first instinct and don't go back and don't change it. You can't go back on multiple choice. That's really a no-no. All right. So what I wanted to review a little bit, because I know that chapter 9 is not as long, but it's a little complicated to really, I think, visualize and understand really what it's all about. So it's mostly parasympathetic and sympathetic, right? And we know that we have the flight or fight for the sympathetic, and I call it the eat and relax for the parasympathetic. And you have a chart on your textbook that it tells you what each organ is doing for each of these two subdivisions of the autonomic nervous system. And I don't want you memorizing that because it's very hard to remember exactly that sympathetic vasodilates the pupil, but in other things, it will contract muscle. So it's kind of like not intuitive to study it like that because it's just pure memorization. So I'm going to tell you how to remember the main system or the main organs and how they react in parasympathetic or sympathetic action, okay? So one of the things that I wanted to do, I have to clear all my doodles here, is remember the reflex arc, like I mentioned, that chapter 9 is all about the motor side of the reflex arc. It's how the body responds to the effectors that are going to be autonomic, right? So from the beginning of nervous system, we learned that the nervous system can also be classified according to the effectors, right? So just the last little portion of our reflex arc. So according to the effectors, we know that we have the somatic nervous system, and that's when the effector is the skeletal muscle. We're gonna be talking all about that chapter 12, right? So that's the somatic nervous system. Somatic means the body, so it helps us move the body, right? Skeletal muscle. And then we have other effectors that are part of the autonomic nervous system. And we mentioned that one is cardiac muscle. We also have smooth muscle, and we also have glands. Now, chapter 11, we're going to talk all about glands. So we're just going to concentrate on that little effector, which are our glands, endocrine and exocrine glands, right? But these autonomic effectors are managed by the autonomic nervous system, so that we have the two sub-branches, the sympathetic and the parasympathetic. So that motor side of the reflex arc, when we talk about the autonomic, is a little more involved than when we have a simple monosynaptic reflex, which will be kind of like the skeletal muscle, right? So for the autonomic, we have that if we're talking sympathetic, you have a synapse on a ganglion. And anybody remembers where the ganglion of the sympathetic is? Anybody remembers the ganglion location?
Isn't it near the effector? Or is it like in the middle or something?
It's along the vertebral column, right?
Right.
Remember the sympathetic is also called craniosacral, right? No. Yes. No, that's the
Yes.
parasympathetic. That's the parasympathetic. Is thoracolumbar. Thoracolumbar is the sympathetic. So we have the sympathetic ganglia along the vertebral column, right? So that the preganglionic is a little shorter. The postganglionic fiber is longer because it has to reach the effector, right? So the ganglia are body of neurons outside the central nervous system. So it means that we have this body of neurons that they are not in the brain, not in the spinal cord, but they're outside, right? So the preganglionic fiber has to synapse in the ganglion, right? So remember, this is my motor response. So we are on the motor side of the reflex arc. So the difference with sympathetic and parasympathetic is that instead of having only one motor neuron going to the effector, we really have two. We have a preganglionic and then a postganglionic fiber. So that's the only difference in the reflex arc, right? In the regular reflex arc, you have integration center, motor neuron, effector. In the autonomic, you have interpretation center, preganglionic, postganglionic, effector. Is that clear? So it's the motor side that is a little different. We still have the same sensory side, no problem with that one. But it's the motor side because our effectors are autonomic, right? So we're talking cardiac, smooth muscle, glands. So in the sympathetic, the preganglionic makes the synapse in this ganglion, right? So the neurotransmitter in this first synapse is acetylcholine. Now, I need to continue with the information to the postganglionic. So what do you think is the receptor on the ganglion for acetylcholine to be able to continue on with the depolarization? Is it a muscarinic or a nicotinic receptor for acetylcholine?
Nicotinic?
Nicotinic, yes. So this little nicotinic receptor here with acetylcholine is going to cause a depolarization on the post, right? So now the postganglionic fiber is a motor fiber, right? We're getting to the effector. Because we're in the sympathetic, the neurotransmitter here is going to be norepinephrine. And we know that norepinephrine is an excitatory type of neurotransmitter, right? So depending on the effector, let's say it's heart, norepinephrine is going to do what? It's going to increase or decrease heart rate?
Increase.
Increase, right? Excitatory, because it's going to attach to a beta receptor in the heart, okay? So that's how you need to study. So you have always acetylcholine on the first synapse. The receptor on the ganglion needs to be nicotinic because we want to make sure that we're sending that information to the postganglionic. And then in the case of sympathetic, we're going to have that the neurotransmitter is norepinephrine. Now, what norepinephrine is going to do on the effector, it depends on the receptor that we have on the particular effector, right? Most of the time, excitatory, right? Norepinephrine is as the same as epinephrine. It's just that in the sympathetic, the neurotransmitter is norepinephrine. Epinephrine is very similar in chemical composition, but we have a lot more epinephrine coming from the adrenal medulla than in the form of a neurotransmitter. Both are the same. As a group, they're called adrenaline, right? But in the synapse, it's more norepinephrine. Now, if we go to the parasympathetic, we're still going to have a we're still going to have a ganglion. Now, this ganglion is located where for the parasympathetic? I already told you. It's called craniosacral, right? So the ganglia are going to be synapsing way up or way down in the body, right? Now, there's a little difference in the length. The preganglionic in this case is long. The postganglionic is short. Some of you said the postganglionic is right on the effector, and pretty much so. They're right on the effector, very close. So that postganglionic fiber is going to be longer. Shorter, sorry. They're close to the effector. So pre is going to be longer, post is going to be shorter because it's right on the effector. So again, the preganglionic neurotransmitter releases acetylcholine. What kind of receptor we're going to have here so that we can continue on with that action potential here? It has to be nicotinic, the same one, right? Because that's the excitatory one. And then at the end here in the effector, the postganglionic fiber is going to secrete acetylcholine again. Now, most of the receptors for that acetylcholine in the parasympathetic are muscarinic, right? Because the parasympathetic, remember, is the eat and relax. So it's more on the inhibitory side. Or I would say not excitatory. So one of the things that I wanted you to think about the sympathetic and parasympathetic is not only that they both have two motor neurons. We have a preganglionic and a postganglionic. They're making a little pit stop or synapse at the ganglia, depending on the location for sympathetic and parasympathetic. They both have acetylcholine on the preganglionic, right? The ganglion is going to have a nicotinic receptor, but the sympathetic will release norepinephrine in the postganglionic. The parasympathetic will secrete acetylcholine from the postganglionic. So the other thing is to think about what are the major organs doing in the situation of fight or flight or eat and relax. So instead of, like I said before, memorizing that table where you have what each of the effectors are doing, and I think it's probably, is it this one? No, I have it. I think I have it on my PowerPoint. Yeah, instead of having all this in your head, which is very complicated, or all this in your head, what is everything doing at each time, right? One of the good things about remembering what your body is doing is just thinking what you need to do in order to fight or flight. So you can't start going through the top of your head all the way to your internal organs, right? So for fight or flight, we know that has to do with the adrenaline rush and that we're trying to fight or to flight, get away from danger, right? But think about, we just mentioned it in vision. Your pupils are going to dilate in the sympathetic because you're trying to see everything. Where is the danger coming from, right? So you want to have as much light as possible coming into your eyes. Then think what happens to your saliva secretion. What happens in your mouth when you're nervous, when you're scared, or
you get dry, right?
Dry. What happens is that you start making a lot more of the mucus saliva than the watery saliva. So that's why you have like pasty or very dry up mouth, right? So that's one of the actions of the sympathetic. Then you start thinking, okay, what's happening to my heart? Obviously, that's a given, right? If you're running or fighting or flying, your heart rate is going to go up. So an action of the sympathetic is heart rate to go up. Think about what's happening with your respiratory rate. Think about what happens when you're running, right? Your respiratory rate is going to go up, right? Because you have to take more oxygen. And then think about your skeletal muscles. What are your skeletal muscles doing when you're fighting or flying? Think about running from the guy with the knife, right? Your
They're
muscles
contracting, they're tight.
They're totally active. And if they're totally active, what do you think they need to be totally active?
Oxygen for the ATP.
Oxygen. And what else for the ATP? We need oxygen and for cellular respiration?
Glucose?
Glucose. So one of the actions of the sympathetic is not only increase your respiratory rate to get that oxygen, but also is mobilizing glucose storage to take the skeletal muscles. So that's part of your sympathetic, right?
And is it increase insulin production?
Yes.
Okay.
Right? Because you want to get more glucose inside the cells, whatever is available, right? So that's how you have to understand the sympathetic actions instead of learning what's going on in each of the organs, because most of our organs have dual innervation, meaning we have a sympathetic fiber and we have a parasympathetic fiber, right? Because remember, as we started talking about nervous system, we need both. You cannot be on high alert 100% of the time, right? You also need the calming down and all that. So that's the way that I want you to learn about sympathetic. And if we go the other way, if you think about the parasympathetic, there's a reason we call it eat and relax, right? Because parasympathetic means that your digestive system is always going to be very active during parasympathetic activity, but the rest is not going to be, right? So think about when it's Thanksgiving and you get that big meal. Some people say we eat 5,000 calories. I can't believe that we do that. But maybe with all the pies, yes. But anyway, after we eat that huge meal, most of us would just sit on the couch and watch TV. At least that's what my family does. So anyway, that's because you have to concentrate all your activity in the digestive system. So there's a lot of blood flow to the digestive system. Your digestive organs are going to be super active. That's under the parasympathetic. That's under cranial nerve number 10, the vagus, right? That's the parasympathetic nerve that innervates the digestive system. But if you think about it, it's parasympathetic, but we're saying the digestive is very active. Yes, because it has to digest the food, right? But think about the rest of the organs. Your heart rate is low. You barely are pushing blood to the brain to be awake, but most of the time you fall asleep. So there's a lot less oxygen going to your brain. You need to put a lot of attention to the digestive system, right? So your heart rate is going to be lower. Your respiratory rate is going to be lower. You're just going to be paying a lot of attention to your digestive system. You're going to have a lot of salivary secretions, right? And in this case, you're going to have both the watery and the mucus saliva, because you have a combination of foods and nutrients that need both in order to make that first bolus, is called, and then treat it in your digestive or your GI tract. So that's the better way of thinking about what does the body need to do to get ready to either fight or flight or to eat and relax. So think about, I don't know, blood flow. How is going to be the blood flow? Well, blood flow is going to be, if you're in the eat and relax or parasympathetic, blood flow is going to be restricted to just keeping your organs alive, because most of the blood flow is going to be in the digestive system, right? So that's why you get a little lethargic after eating a big meal, because there's not a lot of blood flow to the brain. We get just enough so that you don't pass out, you don't go into a coma, although a lot of time people say I'm in a food coma, right? Yes, Julie.
Is that also why some people might feel like when I eat a big meal, I feel cold after? Does the blood flow go to your stomach? And
yeah, and it's not going to your extremities, right? So yeah, everything needs to go right there. When you eat that big meal, we need to manage it, right? And so it's good that we only have that huge, humongous meal during Thanksgiving. We shouldn't be eating that many calories every day. I hope not, right? Just enough to keep us alive. But that's how you have to think, right? Yeah, you want to be on the couch with a big blanket and that's it. Not do much, imagine no blood flow to your skeletal muscles. So don't eat and go exercise. Do all your exercising before the big meal, right? Because most likely if you exercise after that big meal, your muscles are going to suffer because there's not going to be enough blood flow going to them at that time. So that's a, yeah. Julie.
You know, just out of curiosity, I don't eat big meals very often because I don't like that feeling of lethargy. But I do notice when I do, I want, like, it's like I have an urge to take a walk to, like, I guess in my mind, help my metabolism or help the digestion. Is that mental, or does that help the body? I don't
No,
know,
it's,
collect energy.
I think a nice walk is good. I do the same thing. If I ate too much, I'm like, oh God, I need to walk something, right?
Yeah.
And sometimes it's like 8 o'clock at night and my husband says, why would you want to go on a walk right now? And I say, I have to, I can't go to bed like this. So it is good. What I'm saying is don't go play football after that big meal, because exercising that strenuously, it's could damage your skeletal muscles.
That makes sense. But is that the body telling, like, is that the brain telling the body to somehow like generate more energy for digestion, or is that just, you know, an anxious thing? Do you know what I mean?
Yeah, that's a good thing. I don't know, but there must be a connection, I think.
Thank you, Salita.
That it makes you want to move in order to speed up some of your metabolism, I would say.
Yeah.
But there's
Maybe to help like push things through your intestines.
Yeah.
Yeah, I think it's nice and it helps. What it doesn't help is like I used to do a lot of spinning, right, in the club. And I remember seeing some people while they're doing spinning, trying to eat this gel or something, right? And of course, these people are not biologists, that they don't teach physiology. So I would look at them and go, oh God, why would you eat when you're exercising? And cycling is strenuous, right? It's heavy, you're breathing hard. And I think that they're running a marathon, that they need that extra glucose to do this. But in fact, they're probably doing more harm than good, because the minute you put something in your mouth, the systems go, okay, stop blood flow to the skeletal muscles. I have to go digest that. So the blood flow is diverted, right? And that's when you can get hurt. Like, why eat in the middle of the exercise? That's not a good idea. You either eat before, so you have the calories and the energy to get you through, or you eat after, right? But not during. That's what I want to say, because the minute you put something in your mouth, blood flow needs to start being restricted to certain parts of the body. And that's what happens, because we're going to see in chapter 14 that we only have 5 liters of blood total. And there's no way we can give the same amount of blood to every single cell in our body 24/7. We can't. There's not enough. So we have to kind of like play with the sympathetic, parasympathetic, and with restricting blood flow to certain areas while allowing the areas that are active to get that blood flow, so they get the energy and the oxygen. So don't be one of those that eat during exercise. That is foolish. And I could see these people and I would say, oh God, I hope you get injured, because this is not a good thing to do. But that happens. People think that you can do that when you're running a marathon, right? 26 miles, and in the middle of it, you feel like there's no more energy. I only ran a half marathon, you guys, and it's horrendous, I would say. I'd never do it again. So imagine 26. So those people, yeah, because it's such a sustained type of effort that you need a little perk me up of the glucose gel in the middle of it. But an hour exercise on a bike, it doesn't make any sense. You don't need it. If you had a good meal before, you're fine. You can wait until you're done. So All right. Well, your baby is all over, right, Julie? I love it. I
He
love
just learned to wave, so we're like, hi, hi, hi.
Ah.
I didn't realize he was right in the back, but my husband's like, he just did it. He waved.
How old is he?
7 months.
Oh God, that's so great.
Yeah, he's
He's
fun.
got-
He's our first, so everything is like,
Kind of
Very
like
exciting.
independent. He's having fun.
yeah.
I miss that age so much. It was so easy. Now my son is wild.
Really? How old is he?
1.5.
Oh yeah, everybody. I mean, the second he started army crawling, everyone was like, "Just wait, never take your eyes off of him." So I'm getting ready for that because right now he's really just stays put.
Wow, my granddaughter, my second granddaughter is 1.5 years, and I call her little earthquake because she's everywhere. And boy, you can't keep up. I mean, the only time that we rest is when she's asleep.
When they start walking, you can't contain them. That's all they want to do. It's even out in
Go, go,
public,
go.
I have to have someone with me just to go to the store.
Exactly. Oh gosh. Anybody else have little kids here?
Mine are all grown, but I have a friend whose child they visited a couple years ago, and she never slept. The only time she slept was when she passed out, like in mid step. So we just called her
Uh-oh.
Hurricane Amelia because she was everywhere.
Yeah, I love that. But I like it that it's my granddaughter and I just see her and then they go away. Yeah, that's great. All right. So any questions on that chapter 9 type of thing? And how we interpret. So when you read chapter 10, you're going to see that it's all on that sensory side. And then when we go to chapter 11, we're going to be talking all about the glands, the effectors that are also autonomic, right? Because we don't control endocrine glands or salivary glands or sweat glands, anything like that. They're all autonomic, right? Okay, what else? What else can we do? I don't have anything else to do. Questions, you guys? No? Well, I'm
I
gonna.
actually have
Yeah.
one question, but it has to do with the final. Is it gonna, I don't remember how you
It's
teach.
fine.
Yeah, because I like to collect everything, especially now being so busy as a mom. I can't remember if you do cumulative finals.
Oh, no, remember I said my final is, I don't call it cumulative, so you don't have to worry about starting to study from chapter 6. It will be mostly focused on the last 3 chapters, which I think is 18, 19, and 20. But I may have general questions on cardiovascular, respiratory, urinary, because they're so related to each other that I want you to get that connection. You're going to see the connection when we start with blood and cardiovascular. I'm going to start mentioning all those connections. Then we're going to go to respiratory and we're still going to talk about cardiovascular. And by the time we get to chapter 17 with urinary, I'm going to bring back cardiovascular and respiratory. So it's going to be toward the end that you're going to be able to make all those connections. So I think that you'll know those answers because we're going to be relating them as we go. And I'm going to be pointing them out as we go so that at the end you have kind of like what I like you to have, an overview of the human body and how it works and how we keep everything functioning. Just pretty much what physiology is all about. But yeah, you don't have to go and read chapter 6.
Okay, and the study guide would help with that too as well, right?
Yes, because it's just the last 3 chapters and I want you to concentrate on those. And I'm telling you, I think that 19 and 18 are super related. 19 is nutrition or metabolism, which it brings back like cellular respiration, some of the hormones that we're going to learn in chapter 11. So that's 19, pretty much calories in, calories out. And then 18 is digestive, but to me, digestive is one of the easiest chapters. It's very straightforward. It's just one long tube with things secreted into and things absorbing into blood. So very straightforward. So I think that the last 3 chapters are not that hard. I think that probably the hardest are 14, 17, 16. Again, cardiovascular, respiratory, urinary. So the final shouldn't be a problem, I don't think. Should be a lot easier than the others. Good, huh? Good to know. Anything else? No? So are you okay with the amount of work and everything like that? I always like to know to improve things online. It's
So
good.
this is really manageable. This is really
Yeah.
good. Thank you.
Good. I know you had not such a nice experience, right? Before, Karen.
Yeah, this semester is a dream.
Oh, what else are you taking?
I'm taking this class and Chem 202, which I love. I took Chem 201 over winter, and so this is just amazing.
Wow, that's great. Yeah, I loved 201 and 202. Hated organic.
That's next.
So I know some people are the opposite. They love organic and they don't want general chem, but I'm more of a general chem. Oh, Rihanna, you're taking organic right now. Oh, do you like it? Oh, Chris, what is so bad about organic? I don't know. I hated I don't know. I hated the labs in organic. If you're doing it online, it's probably easier, but they were long, tedious. No, I didn't like organic at all. I had to do it, a year of organic for bio majors. We had the worst major ever of all the STEM sciences. A lot of application, many exceptions where general, there's more calculations. Yeah, I like calculations. I like the chemical equations, stoichiometry. I love all that. Titrations, I love that. And not organic, no. Solvents, organic solvents, oh, fuels. I didn't think I would use it. I never did.
I noticed the same thing with physio and anatomy. I'm really good at physio, but anatomy kills me.
Well, because anatomy is mostly memorization. So I think I could memorize something, but it's not as interesting to me as learning a process. Or if this happens, what happens to this? It's always kind of like there's a cause and effect with physiology, right? And if this goes wrong, what happens to that? But with anatomy, it's like point a name and it's, ooh, yeah, they have all their own names. Who's taking anatomy right now? Julie. Julie C, not Julie M. Well, and you know what? It's hard to take anatomy remotely because I think that anatomy it's a lot better for you to kind of like touch things and use the microscope or see the cat. I think it's easier being face to face. So we're doing something for the fall semester, which I think is going to help the anatomy students. Not that you want to know, but we're thinking in bringing some labs back on campus for the fall. You all know that EMT is on campus, welding is on campus, culinary arts is on campus, nursing is on campus. So those are called the essentials. So that's the reason why the CDC allowed them. So their social distance, their body temperature, it's taken. They have to wear a mask the whole time they're on campus. They have to also fill out a health log anytime you go on campus. So it is very, very controlled. Now, on the chat, somebody says, Are we coming back over the summer? I'm confused since high schools are opening. So Chris, no, we're not going back on the summer. Summer is still online for all of us, because the way that the schedule works is that we have to plan way in advance. For example, I'm working as a department chair, I'm just working on the spring 2022 schedule, right? I just work on fall 2021. We have to do everything in advance because we have to have registration open for students. The problem with, so summer was decided we're going to stay remote because we had to open registration so long ago. And the conditions when we had to decide were not the conditions that we have today, right? Things are getting better, vaccinations are going on. All of us in education have gotten vaccines. So it is getting better, but still the CDC has not released or has not relaxed, I would say, the 6 feet social distancing, the mask using and things like that, especially in California. Now you go to Texas, it's another story. No mask, no nothing, everything is open. Same with Florida, right? But here we're being more careful. So summer, it had to be decided a long time ago, and that's why we're remote. For fall, we're still taking it like the guidance. We need 6 feet social distancing and mask, but we can maybe do a little different and do kind of like a hybrid way, just like a lot of schools are doing. Like my granddaughter, for example, she started school, but she only goes for 2.5 hours. She goes in the afternoon. There's another group that goes in the morning. Now we can't do that, but what we can do, and this is what we want to do for anatomy students. So if you're taking it in the fall, this is how it's going to work. We're still going to be online, but the lab is going to be open during the week for students to go and look at models, microscope, cadaver, and there's always going to be faculty there to help you. So that's what we're going to do, because I think anatomy is so visual that you have to have that tactile experience of, oh, what does the liver looks like, not just look at a picture, right? And it's a lot of like 3D for anatomy. So it's a good way of looking at the relationship with the organs instead of just looking at a picture. So that's one of the things that we're going to do. In some other classes like microbiology, we're going to have 3 sections or 4 sections that are going to have some labs face-to-face. So that's going to be good.
So I have to decide whether I'm taking micro or anatomy in the summer. What do you suggest? Because it's been 7 years that I've been in school, and I don't remember how it used to be.
Okay, so, but I need to know, if you take anatomy in the summer, what else are you going to take in the fall other than micro?
It was some easy class.
Okay.
I'm worried about.
Sometimes I say to students, take micro in the summer, but that's the only thing you do in the summer, because in 8 weeks, you're done. It's a long class, it's a challenging class. So it's good to take it as just that class in the summer because you're all done. And that's all you're thinking about summer. It's just micro, right? In the fall, it kind of like 16 weeks is long, and then it's a hard class and you have always something else to take. So I always told my students, take micro in the summer, but do just that because you're done in 8 weeks instead of drawing it out for 16 with probably other classes that you need to take in the fall.
That's what I did. And it was nice to just zero in and focus on that. And I was 35 weeks pregnant. And that's all I focused on. Like, it was a great distraction. And then it was nice to just concentrate on that instead of, like you said, balancing.
That's exactly what I was thinking now that I have my son and it's like, oh, and I'm working, it's a lot.
Yeah.
I didn't even know how I was going to do this semester too. I was so worried.
But I think it's a good idea doing it like that in the summer. If that's the only thing you take, don't put anything, even if it's an easy class, don't do it. Just do that. And then you're done in just 8 weeks. So Chris is asking did it suck taking micro over that period of time, Julie?
I enjoyed it because I had no other distractions, like you said. And it was just an opportunity to, I mean, I just committed. And since it wasn't 16, 20 weeks, it was just 8 weeks of solid micro. I thought it worked out really well. And I was probably, I know I was less distracted than I would have been trying to just go about it. Like, you know, but you have to make that commitment at the beginning, at the top of the class, that you're in it for 8 weeks and that you're going to commit that time and not, you know, set
Yeah.
yourself up for failure.
Yeah, Leslie says that she's taking micro right now with this one. That's a lot, Leslie. I don't know how you're doing it. I hope you're okay and you're not going crazy. Who raised their hand? Yeah, it's really tough. Uh-huh. Somebody raised their hand. Jessica, you raised your hand.
It was an accident.
I did. I'm fully vaccinated, you guys. Woohoo! I got Pfizer. The first shot was nothing, a little sore my arm, nothing. Second shot is a different story. It wasn't bad. I didn't get a fever, but the next day I got my second shot on a Saturday. That's Sunday, and I did it like that on purpose because on Sunday I started feeling like you feel when you start getting sick, like your body hurts everywhere, and I could feel that my hair hurt. And I took my temperature. I didn't have any temperature, but I felt really run down. So I spent the whole day on the couch with a blanket, watching TV. I fell asleep. Then I woke up on Monday and I'm like, nothing happened. It was like a 24-hour thing and I was fine. So no, we can't make the vaccine mandatory. That's a question on the chat. Because this vaccine is still on the trial phase. You know, it's an experimental vaccine, all three of them, Pfizer, Moderna, and Johnson & Johnson. So they cannot be made mandatory. So no, they're not gonna be made mandatory until they are totally, you know, done with all their research. They are testing it in children right now, less than 16. Because Pfizer is for 16 and over and Moderna is for 18 and over. And I think Johnson & Johnson is still. Yeah, so how long does it usually take for it to be out of the trial phase? Oh God, in normal times, 20 years, Chris. This has been sped up really, really fast. So I would say for about maybe next year, something like that, because they still need to make sure that there's no side effects, and we won't know about the side effects for about a year. My sister is a dentist. She's older than me. She's eligible to get it. She doesn't want to get it because she says she's going to wait and see what happens to me. So I'm fine. I still haven't grown another finger, arm, or anything like that. My DNA is protected, you guys. If you hear that this vaccine, at least Pfizer and Moderna, because they're mRNA vaccines, it doesn't get into your DNA, okay? So don't think that's not true. That's a myth. Chris says, that's what I'm doing too. Let me know what happens. I'll check in frequently. Yes, of course. That's my sister. She says, oh, and she said she wants to go to Cancun in the summer. If they say that she needs to be vaccinated, she will do it just to go. Crazy, right? I just want to do it to be protected. That's it. Yes. Yes, Jessica, you can go. We're going to be done with class. We're just chatting. This is good too.
Are you still doing the cadaver and anatomy? Because that was my favorite part.
I know. Yes, we are. We now have 3 in Valencia, and we're going to have 2 in Canyon Country. We're opening the new building in Canyon Country, you guys. So if anybody's taking microbiology or anatomy and physiology in the fall, we're going to have some classes over there, and it's going to be all brand new labs. So we're excited. And we're going to go see it in April. We're going to see the new labs. Yeah, cadaver all the way. Yes. I don't do it anymore because I teach physiology now, but I used to teach 201, 202, 204, 205. I used to teach everything.
Oh, you don't teach 201 anymore?
No.
No.
I don't have time. I can't teach that much. This is my little favorite class. what's the difference? What's the difference between the two locations? Oh, Chris is asking that. Nothing is, we are trying to replicate what we have in Valencia to have a Canyon Country, just because our classes are always so impacted and waitlisted. So that we're trying to offer more sections over there, especially A&P and microbiology. So there's be a lot more sections that we're gonna offer